Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and Inside Scientific webinar titled How to Create CRISPR-Edited T Cells More Efficiently for Tomorrow's Cell Therapies. I'm Liam Sanyo for the events team here at Scientist.com, and I'll be the host today. This webinar has been sponsored by Stem Cell Technologies, so big thanks to them for helping to make this webinar possible. Today, we're being joined by Ian Foster, Regional Sales Manager in Immunology at Stem Cell, and Stephen Loo-Yong-Kee, Scientist in Cell Biology and Immunology at Artisan Bio. Ian will start things off today by giving a brief introduction about stem cells technologies, and then Stephen will present the STAR-CRISPR system for optimizing gene editing in cell therapy with a focus on the genetic modification of T cells for cancer immunotherapy. He'll also be presenting Artisan Bio's risk-based systems approach to technology development, including how to implement design of experiments, or DOE, and quality by design, or QBD, principles to optimize and validate any process. Alrighty, and with that, I'm very pleased to hand things over to the first speaker, Ian Foster. All right, Ian, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Thanks, Liam. Stem Cell Technologies is Canada's largest biotechnology company, established in 1993. Stem Cell is a global leader in life sciences research, serving both academia and industry professionals. We have locations in North America, Europe, as well as Asia Pacific region. In addition, we serve the rest of the market through distributor sales. In terms of the research areas and applications that exist at Stem Cell Technologies, these can range from pluripotent stem cell biology, neurobiology, pulmonary, to areas like mammary and prostate research. One of the core research areas we support is immunology research, which we will be focusing on today. Within immunology, we have a wide range of products that help support workflows for scientists performing immune cell-based research, all the way from sourcing of primary cells to cell isolation, activation, genome editing, cell expansion, and analysis. Isolate the immune subtypes or obtain them directly so they are ready to go when you are. Differentiate, activate, expand, or edit the cells based on your needs. Analyze samples to generate key findings or cryopreserve your samples downstream. The data can be used to create new cell or immune therapies that can help save lives. I'll now pass it on to Stephen Loo-Yong-Kee.
Thanks, Ian. Good morning, everyone, or good afternoon, depending where you're watching this from. I want to thank everyone for coming to join this webinar. My name is Stephen Loo-Yong-Kee, and I'll be presenting how we went about creating the next generation of CRISPR-edited T cells. Before we jump into things, just a little bit about me. My background is actually in engineering, so I got my master's at the University of Toronto in bioprocess engineering, and I had my first job in the same field. It wasn't until I got to CCRM where I pivoted into cell and gene therapy, primarily focusing on immunotherapies, viral vector manufacturing, as well as non-viral transfection technologies. And it's been over 7 years since I first started in cell and gene therapy, and I'm now at Artisan Bio, where we're developing this editing platform that I'm presenting today. I'm primarily focused on Artisan's T cell and iPSC editing platforms, but also giving some insights into DOE and QBD methodologies. I also want to introduce us, Artisan Bio. So we have an office here in Toronto, as well as in Louisville, Colorado. And we pride ourselves in being a one-stop shop for any partners. So what this means is we provide both data and know-how to partners. And data comes in the form of guide sequence data packs, as well as off-target data packs, to look at both efficiency and safety. And know-how comes in the form of protocols and licenses that gives our partners full FTO for their products. And this is all really based on our STAR-CRISPR platform. So one of the key features of our STAR-CRISPR platform is our STAR guides, which is designed with this dual-piece guide design. So the design allows for a wider guide design space, as it allows for tunable activity based on different split locations in the guide based on our partner's needs. And this STAR guide, in combination with our STAR nuclease, kind of makes up our STAR-CRISPR 1.0, which is available now, with 2.0 in optimization studies. So looking at the CRISPR landscape, Cas9 is the most prominent nuclease being used. That's because of its high on-target editing efficiency due to its PAM sequence being quite prevalent in the human genome, which allows you to design guides for pretty much any target. However, this comes at the cost of higher off targets as well, so decreased safety. On the other hand, you have type 5 nucleases like Cas12, 12A, which have better specificity because of its 4 nucleotide PAM sequence. So it has better off targets, so improved safety, but it comes at the cost of lower performance. Using the STAR nuclease, though, we're able to get kind of the best of both worlds. So we have the high on-target performance of Cas9, but then the lower off targets of type 5 nuclease, with a 40% reduction in our off targets compared to wild-type single-guide RNA. And this is due to modifications like novel nuclear localization tags, as well as other modifications that improve on targets. So this STAR nuclease in combination with STAR guides also gives us complete FTO compared to the other two. And we're always looking to improve our platform and our system in order to strengthen our worldwide FTO position. So we've developed this novel CRISPR editing system that we know works, but we want to be able to hone it for each potential cell therapy application that our partners might need. So the way we did this is by taking a systems approach. So a systems approach is really just a big picture view of the whole system, looking at each of the different individual components and how they interact with each other in order to affect the system's behavior. So traditionally, when you're designing an experiment, you might do something like a bottom-up approach or something that's hypothesis-driven, looking at an individual component and testing the hypothesis on whether that component affects your system. On the other hand, you might be taking a top-down approach where you look at large data sets and you analyze that data in order to find patterns to find the components that matter the most. What a systems approach does kind of combines the two of these together and applies both a bottom-up and top-down approach, depending on what stage you're at in the iterative design. So the first thing you want to do with a systems approach is define your solution space. So in the top right, this is kind of illustrated using this diamond shape. And so the solution space is all possible combination of all your factors. So you can think of each axis as a different variable that you can increase or decrease to move through the space. And so your solution is somewhere in that space. And so what you want to do is reduce that space in order to have something more manageable. So from all possible combinations to something that is closer to what your final solution would be. And you could do something like this, and you would accomplish something like this by doing something like a risk assessment or a screening DOE. So now that you have this smaller, reduced solution space, you then want to be able to characterize it and understand all the different factors and all the different inputs and how those affect your final output. And in order to do this, you would need something like a high resolution DOE to kind of model that, what we're calling our design space inside the solution space. So understand how each of the different components impact your system's behavior. And then you'd obviously have to validate that model to make sure that you can consistently and reproducibly get the final product that you want. If this sounds familiar to you, you might have heard of quality by design, or QBD, because QBD really emphasizes product and process understanding by taking a systematic approach to development. So traditionally, you might have some understanding of your inputs or your outputs. So your inputs might be your reagents, materials, and equipment, and your output is your final product. But your process ends up being this black box that's not fully understood. What QBD tries to emphasize is using this systems approach to really understand your process and how the interconnectedness of all your different inputs, so including your materials as well as your process parameters, and how these affect your outputs. So it's really about building in quality from the very beginning by kind of demystifying the process itself. So by taking the systems approach, an iterative risk-based assessments and DOE screenings, we're able to kind of start identifying critical material attributes and critical process parameters as your inputs that affect your final critical quality attributes of your final target product. So bringing this into practice, looking at an allogeneic CAR T therapy workflow, you can start to see some of the QBD challenges here, which are primarily around being able to reproducibly produce the final product. And this is already hard enough as it is, because with a product like a CAR T, which is a cell therapy, cell products are notoriously hard to define the quality target product profile because it's not fully understood and they're quite complex. On top of that, starting from the very beginning, you have variability coming in from your starting material, which is donor material, and so you have different quality and quantity amounts to be considering. Then throughout your manufacturing process, you also have variability because there are several complex steps along the way, such as isolation, gene editing, and expansion, which again introduce more variability to your process. So in order to kind of address all this, there needs to be a focus on understanding the process and understanding which factors are critical to getting the quality target profile that you want. So we want to address some of these issues as we're developing our editing platform for T cells by starting to apply some of these quality concepts into our editing platform development. So similarly, we're looking at inputs and outputs, but instead of looking at critical material attributes, we're looking at controlled and uncontrolled variables. So this includes our reagents, our materials, and our equipment. And then for our unit operation, we're looking at our editing platform and looking at different process parameters, so different levers and gears that we can turn and pull in order to impact our final output, which is our intermediate quality attribute instead of the critical quality attribute. So since we don't have a final product in mind, our output is really just the analytics which feed into our intermediate quality attributes, which is our editing efficiency. So when looking at developing our process, the first step is to really outline the knowledge space. And this space is a total understanding of the process and includes all the possible factors that could impact our process. And so it's a very large space. And at this point, you could perform something like a risk assessment to determine which factors we actually want to test that we believe will have the most impact. And so from there, we move from our knowledge space to our solution space, which is a subset of the knowledge space. And in the solution space, instead of it being all the potential factors that are considered, it's just the factors we've defined, but then all possible combinations of those factors. At this stage of the solution space, we then take something like a top-down approach to do a screen design to collect a large amount of data. And then we would analyze this data to identify patterns like main effects and directionality in order to start understanding our process and defining the boundaries of our solution space and reducing it. With this reduced solution space, we then want to start figuring out what the optimum point would be. And this would mean we would run something like a response surface curve or mixture design DOE to start developing predictive models for identifying the design space. And as I mentioned before, the design space is this very well-characterized space where we can now consistently produce our desired outcomes. And so once we characterize these models for robustness and variability, we now have this design space. Then moving from design space to control space, we would look at understanding which factors need to be tightly controlled in order to consistently be hitting our desired high-quality outputs. And so as we're narrowing down the space from knowledge space to solution space, design space, control space, we're starting to gain process understanding, which we then have to optimize, characterize, and validate before we ultimately land on our set point. So now applying this approach back to the development of an allo CAR T, starting with kind of knowledge space, we did a market research. And what we found was that there was a common medical need for similar edits across the field. And this included multiple payloads, including dual and tri-CAR T cells, in order to address low antigen expression on solid tumors, tumor homing to overcome the microenvironment around tumors that were immunosuppressant, as well as immune evasion in order to increase persistence. So by following the steps that we had laid out previously, we're hoping to develop this process in order to demonstrate high editing efficiency for our partners in order to produce the cells that could address these medical needs. So after doing the market research, what we landed on was this multiplex build, which we thought would be a good case study for demonstrating high editing efficiency in T cells that could be used by our partners. So this build includes a CAR, which targets antigens that are highly expressed on solid tumors, chemokine homing, which enables a CAR T to home towards the tumor and increase efficacy, as well as an allo chassis, which includes knocking out the TCR, HLA-1, and HLA-2 in order to prevent GVHD, and enabling the allo off-the-shelf product use. So the first step was tech transferring our existing T-cell editing process. So ahead of this project, we had actually done an evaluation before looking at commercially available reagents that were either GMP or with a clear path to GMP that kind of fit to help develop this workflow. And what we found was using stem cell technology's suite of T-cell products best fit our needs. So the first step of this T-cell workflow is designing our STAR guides. We then sourced frozen leukopaks from stem cell technologies and isolated T-cells using their AZCEP T-cell isolation kits before freezing the cells in Craftdoor CS10. The cells were then expanded in Immunocult XF media with IL-2 and activated using Immunocult CD3, CD28 activators. We then would prepare our RMP, which is a combination of our STAR nuclease and our STAR guides. And then after delivering the RMP to the cells, we then expand them further, again in Immunocult XF with IL-2. At the point of harvest, we then do our downstream analytics to determine our editing efficiency in the form of flow cytometry, which would give us % knockout and knock-in based on marker expression, as well as our orthogonal method of amplicon-seq to determine % indels. We were also interested in functionality of our CAR knock-in, and so we were also looking at cell killing assays to determine cell potency. And we still wanted to make sure that our editing system was safe, and so we would look at off-targets using RAMP-seq. The next step was to perform a risk assessment. So we broke down the workflow that on the previous slide into major unit ops, and we had a group of SMEs kind of come up with a laundry list of potential factors that they thought might impact our final intermediate quality attribute, which is our editing efficiency. And so at this point, you can kind of think back to this is our knowledge space. So this is everything we know about our process to date. We then had that same group of SMEs rank each of the factors from highly likely to very unlikely, so our 3, 2, or 1, on whether they thought this would impact either knock-in efficiency or knockout efficiency. You'll also see some of these factors got grayed out, and that's because of the group of SMEs. It was decided some of these factors were out of scope for the body of work and just to really focus the experiment as well. So now that we had rankings for each of the factors for both knock-in efficiency and knockout efficiency, we would multiply the two of these together in order to get a total rank for that factor. And then looking at the total ranks for each of the factors by each SME, we're looking at the factors that are consistently at the top in order to promote those ones to our DOE. With this, we've kind of defined our solution space now. So next was actually design and execute our experiment. So we opted for a definitive screen design because we were interested not only in which factors were kind of the main factors or critical factors, but we're also interested in understanding the interactions of each of these factors, kind of back to that idea of a systems approach where we want to understand the interconnectedness of the factors. So we kept some things constant throughout this workflow. So we still use frozen leukopaks as well as stem cell technologies, T-cell reagents throughout this entire culturing. And we also only looked at a single transfection method for this experiment. From the very long list of factors on the previous slide, we actually only came up with 6 factors that end up being the top candidates for what we believe would be impacting our editing efficiency. And with this DOE design, that came up to 99 individual runs that had to occur, not including controls. So we went about executing on this experiment. And so we followed the workflow as laid out below, and we performed some analytics along the way. So we looked at viability, cell density, and phenotype throughout the process to understand how our editing was impacting these metrics. We were also interested in cell cycle to see how that would impact our editing efficiency. And last but not least, we had our key readout, which was editing efficiency, which would feed directly into our intermediate quality attributes of knock-in efficiency and knockout efficiency. So with all this data, the next step was now to fit the model to the data to determine any patterns in the system. So with knock-in efficiency, what we found was that most of the factors within the solution space we were looking at were not significant. But what we were able to do was actually find one factor that was very, very significant for knocking in. And with this model, we had very good model fit with 0.89. So it seems like we were able to identify one main factor, which helps reduce our solution space. Looking at knockout efficiency, with this first DOE, we were able to find that, so factor 5, you can see it was not very significant, but looking at factors 1, 2, and 3, you can see that when you start going to lower levels of each of these factors, we're starting to see a drop off in our triple knockout efficiency. Similarly, with factor 4, we're starting to see a drop off at high levels of this factor. So what this is allowing us to do is go from this larger diamond that we have to the smaller diamond where we have a smaller bounds that we're interested in studying, because this is where we're getting the highest editing efficiency. And this is only the first iteration of DOE, but we're already able to start reducing our solution space, and I'll start identifying main factors. And with this initial data set, we're already starting to demonstrate really high editing potential, which means that we believe we're on the right track, because we think the solution is probably somewhere in the solution space that we've identified, since we're already getting upwards of 90% knock-in, which is quite comparable to what competitors are showing with Cas9. And similarly with knockout, we're able to get over 90% knockout, which is again comparable to what's in the field. So the speed at which we're gaining this process understanding from a single iteration kind of demonstrates how quickly we've been able to optimize for different cell types. So in this presentation, we've only really covered the process understanding that shows us what factors are important and started to narrow our solution space. And the next steps would be optimizing, characterizing, and validating this process to kind of define our set point. However, the rest of this process development would really be product specific, and we would kind of take these on with a partner in order to set proper targets that we could work towards. However, the process understanding that we've shown today is kind of the foundation or the building block upon which we would work with collaborators to kind of build these products. And we just want to show that a systems approach is widely applicable across a range of cell types, not just T cells. And we've also demonstrated that in-house as well. So by using this approach with HSCs, multiple factors were identified in DOEs as being critical for improving editing efficiency. And early data is already showing that we're able to get 78% knockout and 27% CAR knock-in while maintaining CD34 percentage. And we're especially proud of our iPSC data, which also incorporates stem cell technologies reagents. So using this iterative DOE approach, we were able to identify multiple factors that positively impacted knock-in efficiency. So we went from having a knock-in of only 3% to 13% to 29% to now 55% using the STAR-CRISPR platform in iPSCs. And this is only preliminary data, and we're also able to show over 90% knockout of HLA-1 in iPSCs. So summarizing the systems approach, we've taken the QBD principles and kind of applied them to our process development. This approach is iterative, as you're kind of seeing with the iPSCs, but DOEs are the fastest and leanest way to great results. And this has been deployed with great success within Artisan. As the pace we've gained process understanding across cell types has really allowed us to quickly improve editing efficiencies and enabled rapid development of these baseline processes that we can then work with partners. So all this process understanding is wrapped into Artisan's know-how in the form of protocols and builds for our partners that are available for licensing. And lastly, I just want to acknowledge everyone that helped out with this project, particularly the team members that are in bold with who made this all possible.
Fantastic. Well, thanks so much, Stephen and Ian, for the great presentations today. And with that, we'll move right on to the Q&A. And so I'll invite Stephen and Ian to jump back on the line here. Well, let's start off with a question that we got from registration. What do you think is the biggest challenge that you've seen thus far when CRISPR editing iPSCs?
I think the biggest one that the team has seen here is really just recovery post-nucleofection. So ensuring that the cells are healthy and viable afterwards. I think it's definitely been the biggest challenge. And we can get high editing, but sometimes you don't always get the best recovery. So it's kind of a good balancing act, figuring that out.
Fantastic. I'm not sure if you have much experience with this, but is there anything that you've seen from stem cell side?
Not something I've seen personally, but from talking with some groups, definitely, yeah, the knock-in efficiencies can be low for sure. I think kind of Stephen touched on that a little bit, and off-target effects are still an issue that a lot of groups are looking at.
Excellent. Yeah, thanks a lot. All right, another good one here. What is the impact of starting with fresh versus frozen samples in terms of editing efficiency?
So this is actually something we did look at in the past, and we've tried, both fresh leukopaks that were frozen, as well as frozen leukopaks that were also refrozen, and we haven't really seen a big difference in editing efficiency using either of the starting material.
Fantastic. All right. How are you designing guides to mitigate off-target effects?
So this is a question that probably be better answered by our NGS team, but from the best way I can describe it, from my understanding, is we have I believe it's we use software to determine potential off targets, and so to nominate guides that have the lowest hazard score, and so that's how we're kind of designing our guides to reduce potential off targets.
Excellent. Yeah, great answer. A question that came in from registration. So this person said, ensuring that the CRISPR engineered cells are perfectly devoid of any transformation associated functional phenotype, or TAFP, compared to wild type is oftentimes impractical in terms of cost or time. But Stephen, what's your approach to ensuring a probable absence of a TAFP while keeping the process in rational bounds?
So this is something we've looked at. So I guess our big focus is genome integrity post-editing. And so we look at things like karyotyping with G-banding, as well as, again, off-targets using RAM-seq and also different pan-cancer panels. But I guess if time and money is an issue, then we also primarily look at different phenotype markers that are available.
Fantastic. All right, another question here. Does use of your process require a CRISPR license?
So our platform actually has it's separate from CRISPR. So we have our own licenses that are available for our STAR-CRISPR platform. So it's different than your typical Cas9 licensing.
All right, another kind of similar question. Is it possible to buy just the STAR nuclease and guides?
That would be a good question for our BD team. I'm not exactly sure what the licensing, I guess, looks like right now.
Fantastic. Not sure if this is information that you can share, but Stephen, how do you perform the knock-ins? Is it HDR induced knock-ins, and how long is the template?
So we do use HDR induced knock-in. We tried different HDR templates as well, and in our hands we've seen different templates work as well. In terms of length, it really depends on how big your insert is, but we have looked at dual and tri-cars inserted, so they have gone quite large.
Excellent. A question about the DOE and systems-based approach. Somebody's asked where you might go for resources in helping to build an approach like this. Is there somewhere, or maybe online resources or something that you can share where people can sort of learn more about how to do this for themselves?
So we've been primarily using Jump. So I think Jump has a great community forum that you could maybe leverage as well. But I guess the best way is just trying it yourself.
Excellent. All right. Beyond a knockout or knock-in transfection efficiency, what additional cell quality attributes do you think are most critical?
I think definitely cell health. So we kind of measure cell health through viability and fold expansion, and making sure there's nothing kind of weird going on in terms of our expansion kinetics. So yeah, I think cell health is definitely one big one for us.
Excellent. All right, another good question. What's the payload size possible for knock-ins?
I would have to ask our Artisan Bio team the exact, but the biggest size they've tried is. So yeah, sorry, I have to get back to you offline.
Yeah, this is certainly I was just going to say, as I was saying earlier, we definitely can follow up offline for anybody where we don't have the answers right away now. All right, and I think we just have one more question here. And I think it's a nice one to sort of close us off here. Where do you see the biggest developments in the gene editing field going in the next few years? And so, Stephen, maybe we can start with you, and then Ian, we can get your perspective.
Yeah, I guess the biggest thing right now that we're noticing is a shift away from viral transductions. That was really popular back a few years ago now with Kymriahs and Yescartas. And a lot of people are moving to these CRISPR-based editing platforms. So I think that's kind of what seems to be where the field's moving towards. And that's why we developed this STAR-CRISPR platform in the first place, try to aid in that.
Yeah, I think I would agree with Stephen. And in general, I think we're seeing also a movement of groups, a little bit like Stephen touched on, looking at PSC-derived immune cells as well, and editing those in addition to primary immune cells.
Very cool. All right, thanks so much. All right, and with that, I think we'll close things out. But thanks so much, Stephen and Ian, for sharing your insights today. It's really been a pleasure having you with us.
Thanks for having me.
Thank you.
Yeah, big thanks also to the audience here for participating. And in closing, we hope you enjoyed this Scientist.com Inside Scientific and Stem Cell Technologies webinar, and we look forward to having you with us again soon. Have a great day, everyone.